To my recording of the labs. Okay, let's go ahead and get going. Right now, first thing I'm going to do is I'm going to show you the three animations of the things that, the kind of big stuff we're trying to get across. Did everybody get a chance at a cookie?
Cookie? Too late.
There's a few Thin Mint Girl Scout cookies. I figure I could leave them for the faculty or I can give them to you. So there you go.
Thank you to you.
Yeah.
That was very thoughtful. These times I said it first.
Yeah. Is that lit up there? Is that working?
It looks like it is. Maybe it's broken, we just need to go home.
Yeah.
He's a homer.
In glycolysis are the additions of two phosphates to the glucose molecule at the expense of cells derive energy from the oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis.
This is what we've
The
gone
energy
over already.
released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell. The initial steps in glycolysis are the additions of two phosphates to the glucose molecule at the expense of two molecules of ATP. The result is a 6-carbon sugar diphosphate.
So what's happened here is this is taking place in the cytoplasm. This is taking place in the cytoplasm of the cell, not the mitochondria. You've got your glucose, it comes in, it takes 2 ATP, are donating their phosphorus. This is not, the batteries are going down. 2 ATP are donating their phosphorus, you're breaking the bonds, you're adding the phosphorus to the 6-carbon glucose, so you have a 6-carbon diphosphate, 2-phosphate. Yeah.
What does ADP become when it loses the phosphate?
ATP, when it loses a phosphate, becomes ADP.
But
It started as ATP. Hang on one
Okay.
second. So you started with 2 ATP. The phosphates come off the ATP, they go to the 6-carbon glucose, and you come up with the ATP goes to ADP.
Would you call it glucose diphosphate?
No, because it's stretched out, it's changed. There is a term, it's not coming to mind right now. Let's get through this until I
Expense
decide
of
to
two
stop.
molecules of ATP. The result is a 6-carbon sugar diphosphate molecule and 2 low-energy adenosine diphosphate molecules, or ADP. This 6-carbon sugar diphosphate molecule is then split into 2 3-carbon molecules. Each of the 3 carbon molecules is converted through a series of steps to pyruvate. During these reactions, electrons are transferred to the coenzyme NAD+ to form NADH, and ATP is formed. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted. Under aerobic conditions, the pyruvate is further.
So you go down to here, it's broken into 3 carbon, you've got pyruvate. Right there. The phosphorus are taken off. NAD is reduced to NADH.
And ATP is formed.
Remember what we had up here, oil rig. Oxidation, lose, reduce, gain. So what's happened is your NAD in there has picked up a hydrogen, it's been reduced to NADH. And that's very important.
Formed. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid.
Okay, so this is stuff that we went through. Pyruvate. You add the 2 hydrogens back, you end up with lactic acid. If it stays in an anaerobic where you don't have oxygen at the end to accept those hydrogens and form water, it stays there and forms lactic acid that builds up in the muscle. Now there is one slight advantage. In the time that it takes a molecule of glucose to go through the Krebs cycle twice, creating maybe 30 to 32 ATP, the pyruvate, not very efficiently, takes 32 molecules of glucose and forms 64 ATP. So you get twice the amount of ATP in the same time frame. That's anaerobically. It can't go on very long. The lactic builds up, it gets poisonous, you get sore. It's not a long-term solution, it's a very short-term solution. But you can get a lot of ATP at the expense of a lot of glucose very quickly. Twice as fast as a Krebs cycle. Just throwing that in because I believe Mary has something like that in your supplement.
But it takes more glucose.
Takes 32 times the glucose. And then, then you may get some of it back, goes to the liver. Remember we talked about the Cori cycle where the lactic acid gets to the liver, it's converted to pyruvate, the phosphates are taken off, it goes back into glucose 6-phosphate in the liver. Glucose is released from the liver, goes back to the muscle. That's a Cori cycle. Lactic acid out of the muscle, into the liver, change to pyruvate, change to glucose 6-phosphate, change to glucose, back to the muscle. There's a circular thing there.
As a way to draw that from the board.
Well, we'll go through it. It's in the other thing.
Is the Cori cycle slow? How can it get rid of all the lactic acid?
They say 48 hours. If you have a big lactic acid buildup in your muscle and the glucose and glycogen are depleted, they'd say it's like 48 hours to get it back. But you had asked me last Tuesday about exercising, and as you exercise, your muscles do gain the ability to get glucose from the fatty acid, more of an ability to get glucose from the fatty acid chains. And as long as there's oxygen, that just provides a lot more glucose before it goes into, it's through the Krebs cycle, before it goes into the lactic acid cycle.
So less lactic acid builds up.
But as lactic acid builds up, you get sore. That's one of the big things about making your muscles sore. Okay.
Cells derive energy from the oxidation of nutrients such as glucose.
Okay, that's the That's glycolysis.
During glycolysis, glucose is broken down
This
to
is the
pyruvate.
Krebs cycle. This is a biggie.
A 2-carbon fragment of pyruvate is used to form acetyl-CoA. The acetyl-CoA enters the Krebs.
Okay, I'm using these a little bit differently because this actually goes very well. So you've got, I wish I had my laser pointer working. You've got pyruvate. You've got acetyl-CoA. The pyruvate is changed into acetyl-CoA. Now it gets into the mitochondria. Now you're in the mitochondria. The acetyl-CoA is a 2-carbon with a 2-carbon molecule. It combines with, I want to say a glutaraldehyde, it combines with a 4-molecule carbon in the Krebs cycle. Now, this is where you need to get your head around it a little bit. The 4-carbon molecule that it combines with just circles around in the Krebs cycle and carries this acetyl coenzyme A to the 7 or 8 steps where it's acted upon, all the energy is released, then it gets back up to the top, picks up another acetyl coenzyme A, and goes around again. Okay? We'll see it when it happens here, and I'll stop it again. But you've got a 4-carbon molecule that picks up this acetyl coenzyme A, acetyl-CoA, and carries it through the Krebs cycle. It's decarboxylated, the hydrogens, it's oxidized, all this energy is pulled off of it, and in the end, you end up with CO2 and water, and this 4-carbon molecule is there to pick up the next one that comes in. Now, it takes 2 Krebs cycles to oxidize or to fully utilize 1 glucose molecule because you get 2 pyruvate. It's 1 pyruvate that makes acetyl-CoA plus a CO2. The CO2 is one of the byproducts of metabolism, and it's what you exhale. So let's go on.
So now we're not in the cytoplasm.
You're in the mitochondria now.
Yeah.
You're in the mitochondria. Now remember, from glycolysis, you got 2 ATP. They're floating out in the cytoplasm. You got 2 NADH. They are in the cytoplasm. And we'll touch on it a little bit later. But the NADH in the cytoplasm don't supply as much energy as the NADH from the mitochondria. You actually lose a little bit by the time they get the electrons into the mitochondria, with rare exception, and I'm not going to waste a lot of time on it. But, so that's why the numbers change a little bit. But let's go on.
Krebs cycle, which occurs in the mitochondrion.
Okay, we'll start.
A 2-carbon fragment of pyruvate is used to form acetyl-CoA. The acetyl-CoA enters the Krebs cycle, which occurs in the mitochondrion. During the conversion of pyruvate to acetyl-CoA, carbon dioxide, CO2, is produced, and a molecule of NADH is formed.
There goes one of your
The
carbons.
2-carbon acetyl portion of the
Now you're down to a
acetyl-CoA
2-carbon.
is transferred to a 4-carbon molecule, producing a 6-carbon compound.
The 4
The
carbon
CoA
carries
carrier
around.
molecule is released. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to NAD+ to form NADH. Next, a second oxidation and decarboxylation occurs. Again, NADH and carbon dioxide are produced. In addition, a molecule of ATP is produced. As a result of these reactions, a 4-carbon molecule is formed in the Krebs cycle. Finally, the 4-carbon molecule is further oxidized, and the hydrogens that are removed are used to form NADH and FADH2. These reactions regenerate the 4-carbon molecule that initially reacts with acetyl-CoA.
So what's happened is, I know, we'll run it again. You gotta look at it a couple of times. I would encourage you to go on YouTube, look up this reaction, You'll get it this simple, it's as complex as you want. But again, here's the there's the four, I'm bringing my own bag. I've got them in my briefcase, but we're not going to do it tonight. There's the carbon molecule. It literally just keeps carrying that acetyl-CoA around the Krebs cycle. It picks one up, carries it around. By the end of the Krebs cycle, the acetyl-CoA is all used up. The 4 carbon molecules there, it picks up another one and starts the circle again. And just keeps going round and round and round. Now this is important because when you start breaking down your carbohydrates, your fats, your proteins, your nucleic acids, what goes toward the Krebs cycle is pyruvate. When you start reprocessing your lactic acid, what goes into the Krebs cycle is pyruvate. Everything comes back to pyruvate, with the exception of when you break down fats. If you have a long chain fatty acid, and we'll get to this, but you've got a long chain fatty acid, say 16 carbons. It's got an acetyl group on the end. It gets chopped off in a 2 carbon chain. You've got essentially acetyl-CoA. So it's, as you're breaking up this long fatty acid chain, for each 2 carbons, you run one of these cycles. And so if you're breaking up a 16 carbon, you run this cycle 8 times as opposed to twice with a molecule of glucose. That's why you can store so much energy in fat. Fat gives you 9 kilocalories per gram. Carbohydrates and proteins give you 4. So little over twice as much energy in a gram of fat as in a gram of carbohydrate or protein. This is why. The fat breaks off into 2 carbon chains, and you've got, they're coming off as acetyl-CoA, going right into the Krebs cycle, going through the Krebs cycle. A 16 carbon chain of fatty acid will give you about 108 ATP as opposed to a molecule of glucose that goes through Krebs and through the electron transport system. Both of them go, the entire total is 108 for the fatty acid chain, 32 for the glucose. So that's why your body is storing your energy in fat. It's just a lot more efficient. Okay, let's go on.
Each glucose molecule is broken down into 2 pyruvate molecules during glycolysis. Then each pyruvate is converted to acetyl-CoA, which enters the Krebs cycle. Thus, for each glucose molecule, the Krebs cycle must complete 2 circuits to completely break down the 2 pyruvate molecules.
Okay, now what happens? You've got this ATP, that's done. That floats out of the mitochondria, it's into the cytoplasm, it's into organelles that use it, it's into the nucleus, it goes wherever it needs to go. But you've got all this FADH and NADH. What you've taken, remember when we talked about the laws, the second law of thermodynamics, if it becomes more structured, you're storing energy in those bonds. When it becomes less structured, when it breaks up, you're releasing that energy. First law of thermodynamics, energy cannot be produced or destroyed. It just changes shape. For instance, you have CO2 and water. It takes energy from the sun. It makes carbohydrates, makes glucose. It makes it much more strong, much more structured. Instead of having these molecules floating out there, you're in a bread. They're in a plant of wheat. They're in alfalfa. Excuse me. They're in alfalfa or they're in whatever. Much more structured. This means that those bonds that are holding that contain energy. When we break those bonds, when we digest the wheat, we are releasing that energy as glucose or carbohydrates for the body to use. The energy is stored in those bonds. That's where we're going. So now you've got the 2 ATP, but you've got all this NADH and FADH. There is a lot of energy that is stored in those hydrogen bonds. When you have reduced those things, because you've taken the hydrogens again that are floating out in the cytoplasm, attached them, you've put energy in. You've made higher energy bonds. Let's see how we get it out. Now this is the electron transport system. This is where your NADH and FADH go from the Krebs cycle.
During glycolysis and the tricarboxylic acid cycle, oxidation of organic molecules results in production of reduced coenzymes such as NADH. These coenzymes transfer hydrogens to the electron transport chain, which is located in the bacterial cell membrane. A hydrogen consists of a proton and an electron. The electron transport chain consists of a series of special electron carrier proteins that shuttle electrons from NADH to a terminal electron acceptor, such as oxygen. Electrons enter the electron transport chain when NADH transfers its protons plus electrons to a membrane-embedded carrier protein. The electrons are sequentially carried along the electron transport chain, while the protons are shuttled to the outside of the membrane. Some of the electron carriers, such as coenzyme Q, accept a proton from the inside of the cell membrane as it accepts electrons. The proton is then transported through the membrane as electrons move down the chain.
Okay, let's go back to this. Remember when I told you we were starting chapter 5, that the electron transport system is kind of like a dam where you're building up these protons, and then you run, like you build up water behind a dam, and then you run it through a turbine. Kind of ring a bell? Right here, let's see if I can get this to work a little bit. That's as much as it's going to work. Right up here is the water behind the dam. The energy, the hydrogens that are coming out, the reduced NADH, the reduced NAD, and the FAD, this is the water that's being put up behind the dam, these protons. See this yellow thing at the end? That's your turbine. What's happened is you're getting a gradient on that side where you have a lot of hydrogen and they're trying to get back into the cell. They're allowed to come through the turbine, and the power that they create, the energy that they create coming through, takes ADP, attaches a phosphorus, and you have ATP. That's where the majority of your ATP in the cell is formed.
And what is that yellow part called?
That's ATP synthase. It's an enzyme and a protein together there. So but you know, I wasn't just making up stories. This is what we're looking at. And I told you, at least in my mind, it's coming together.
The membrane. Some of the electron carriers, such as coenzyme Q, accept a proton from the inside of the cell membrane as it accepts electrons. The proton is then transported through the membrane as electrons move down the chain. This increases the proton gradient across the membrane and enhances the proton motive force. During aerobic respiration, the last carrier protein transfers a pair of electrons to oxygen at the end of the electron transport chain.
Okay. What happens here is all these hydrogens are getting pushed out here. Your electrons are moving down the chain of balls till they get, if you just see the last one before the yellow. Now, the bottom line is the reason we need to breathe oxygen is those electrons attach to an oxygen forming water and they're eliminated. If we don't have the oxygen to attach that to, you might get a little energy out of the lactic, the anaerobic, not out of the Krebs cycle, but everything stops. This blue ball right before the yellow, just to the left of the yellow, is where cyanide attacks. Cyanide stops the electron transfer at that point. And you want to know how fast it is? You're dead in a couple of minutes. That's how important it is. Cyanide will kill you in a couple of minutes. And there's really not much you can do about it. What happens is the cyanide, actually, the brain senses that it's not got the oxygen, especially with the cyanide gas. And the first thing it's going to do is it's going to say, take a deep breath. You don't have enough air. And you just suck in more cyanide. And, I mean, it's just, you know, it's deadly. It's what the spies used to use and all that.
What's the chemical composition of cyanide?
I don't know off the top of my head. You know, I know. In Guyana, Jim Jones's, they use the ferric cyanide, but I'm not sure now. I don't know what the actual chemical structure is of cyanide. I'd have to look at it. But it's nasty stuff, obviously, because they killed, what, 600 people there at the colony down there?
And water is formed.
Okay, I'm going to back it up just a little bit.
Team transfers a pair of electrons to oxygen at the end of the electron transport chain, and water is formed. The enzyme ATP synthase utilizes the energy of the proton motive force to synthesize ATP. This enzyme allows protons to pass back into the cell and couples the energy released in this process to the phosphorylation of ADP to form ATP.
Okay?
Could you go back to that last part, when they were talking about the enzymes allow the ADP to change to the ATP?
Sure.
To me, it's missing a phosphate, no?
The phosphates are floating around in the cytoplasm of the mitochondria, but it only needs one. It's at an ADP, not an AMP. ADP plus one phosphate gives you an ATP. Adenosine diphosphate to adenosine triphosphate.
The energy released in this process to the phosphorylation of ADP to form ATP.
So what happens, and we'll get into it in chapter 6, is you're creating a gradient. And it's a forced gradient because you're pumping these hydrogens out. They concentrate. Now that says outside the cell. That's really not correct. That's in the outer membrane of the mitochondria. This is taking place pretty much in the middle part of the mitochondria. You know, on the mitochondria, you have an inner membrane, a space, and an outer membrane. It's not outside the cell. It's in the cytoplasm. It's just outside the mitochondria. But this yellow thing, the ATP synthase, they feel, and this is on one of the other things that I saw, that you notice how it's got, kind of the banana-shaped structures at the bottom. They feel that actually spins. The hydrogen coming through causes that to spin, and it literally forces the phosphate back to the ADP, forming ATP, pushing it in close proximity and getting the bond. Okay? So this is your electron transport system. This coupled with glycolysis, where you get 2 ATP, 2 net ATP, you get a couple more ATP out of the Krebs cycle, I think 4. By the time you're done with this, you're at 30 to 32 ATP. That's your accounting for glucose. second.
It's like watching TV on the couch.
We should have had popcorn, right?
Just no, it's not.
Okay, glycogenolysis. Glycolysis. Glycolysis, I believe we got through, right?
Yes. You into the aerobic.
Aerobic respiration. Okay.
The aerobic respiration.
We went through lactic acid, we went through glycogenesis, we went through the Cori cycle, we're at aerobic respiration. Okay, cellular respiration. C6H12O6. Glucose goes to CO2 in water. Second law of thermodynamics. Gets to a more random state. When doing that, it releases the energy. The energy gives us NADH and ATP. Then it gives us pyruvate. Like I said, pyruvate for this, I mean, it comes back, it breaks off a carbon, it gives you acetyl-CoA. Acetyl-CoA goes through the Krebs cycle. Okay? So a lot of these things will come back and you end up with pyruvate. The lactic acid pathway through the liver. When the LDH, lactic dehydrogenase, if you remember, like I said, pyruvate plus 2 hydrogen gives you lactic acid. Lactic dehydrogenase takes the 2 hydrogen off, you're back to pyruvate. Pyruvate goes into the Krebs cycle, and boom, you've got your another 25 or 26 ATP. By the time it goes through the Krebs cycle and through the electron transport system. The fate of pyruvate leaves the cytoplasm, enters the mitochondria. Carbon dioxide is formed to remove, carbon dioxide is removed. Acetic acid would combine with coenzyme A to produce acetyl-CoA. Acetyl-CoA is what attaches to that 4 carbon carrier molecule and goes through the Krebs cycle. Take a deep breath. As many as you carry. So 1 molecule of glucose gives you 2 molecules of acetyl-CoA and 2 molecules of carbon dioxide. That's what you exhale. That's the carbon dioxide that you exhale. Okay? Pyruvate, coenzyme A, acetyl coenzyme A. And for the process, you get an NADH. And you saw in the electron transport system, what happens to that NADH? The hydrogen, it is oxidized. The hydrogens are, the electrons are attached back to oxygen. The hydrogens are used to run through the ATP synthase. Krebs cycle, citric acid cycle. Acetyl-CoA combines with oxoacetic acid to form citric acid. Okay, now I'm not going to go into this too much because, you know, the take-home message is the acetyl-CoA goes in, it goes through the Krebs cycle, that 4 carbon carrier carries it around, and you get the NADH, you get the FADH, and you get the ATP. In other words, I'm not going to put on a test what does guanosine triphosphate do. Got that? Abbreviated diagram, let's go to the more.
You're going really fast.
What's that?
You're going really fast. Like, I can't even write this down.
It's all in the text.
Yeah, these are in the text.
Exactly, from the textbook. I'm following it right now. It seriously is like that
Okay,
picture.
Am I getting it pretty close?
It's exactly.
What page is it on?
It's, I'm sorry, 112. Page 112.
Okay. Abbreviated glycolysis, you get the pyruvic acid. The pyruvic acid goes to acetyl coenzyme A. You're in the mitochondria, it goes through the Krebs cycle. You get, for each round, you get 3 NADH. one FADH and one ATP. It takes 2 of those rounds to process 1 molecule of glucose. Okay, that's kind of the take-home message for glycolysis. Then it goes from here, and as I showed you, the electron transport system. That's where your NADH and your FADH are reduced, and those protons are used to create, again, more ATP. The end product is they're combined with oxygen to form water, and the oxygen, that's why it's called oxidation. Your oxygen is a good acceptor of protons, but in the end, you need that oxygen in the aerobic pathway to keep it going, to stay alive. If you don't have the oxygen, it stops and you die very quickly. Some muscles can go long. Muscles can go longer. The brain can use ketone bodies a little bit. The heart a little bit more, but it doesn't last very long. This is the Krebs cycle like you just saw. You can refer to it back in your book if you want. Product of each Krebs cycle, 6 NADH, 2 FADH, and 2 ATP. If you remember, right here, lower right corner, 3 ADH, 1 FADH, 1 ATP. That's half the cycle. That's from 1 pyruvate. Okay, electron transport chain. Do you have any questions on that? You see what I mean? It breaks off the protons, it shoots them up in, stores them outside the membrane. They come back through the membrane. Then with ATP synthase, they power it, they give it energy. It takes ADP, pushes the phosphate up, creates ATP.
You're talking about the last one we just seen, right?
The electron transport system, yes. Because that's where these go. That's where these go. This is the end of your energy producing process in the cell.
So the Krebs cycle,
Can you go back to that slide?
Sure. The Krebs cycle what?
and the electron transport chain together make 32?
Yes. Plus the 2 that you get from glycolysis. So you get 2 from glycolysis and another 30 or so, 28 to 30 between the Krebs cycle and the electron transport chain.
But the 2 from glycolysis are just from 1 glucose, right?
The 2 from glycolysis are from 1 glucose molecule. Yes.
So you don't count those with the...
Well, they're counted in the overall energy obtained. They're floating around in the cytoplasm.
Is it a separate glucose molecule for the Krebs cycle and for the...
The glucose molecule never gets into the Krebs cycle.
It's just
The glycolysis, it breaks it into pyruvate. The pyruvate goes into the mitochondria and into the Krebs cycle, not glucose. Okay? Have you got this? We're good?
Next, thank you.
Okay, now we're back to the electron transport chain, oxidative phosphorylation. It's oxidative because the NADH and FADH are oxidized. They lose the electrons, oil. Oxidation, lose. Oxidative phosphorylation, if it's oxidative, you've got the protons out there. When they come back through, they phosphorylate the ADP to ATP. That's phosphorylation. Electron transport chain, ATP production. This is part of your electron transport chain. The proton pumps are in the membrane. You saw them pumping the hydrogen out. Those are proton pumps. You get a huge concentration gradient, like I showed you, that's a water. The concentration gradient is the water behind the dam. It is just waiting to come through that turbine, through the ATP synthesis, and push, make the ATP from the ADP. This is just essentially what you saw. So you see here, you've got it better. There's your inner mitochondrial membrane, your inner membrane space where they all are, and then your outer mitochondrial membrane. It's not outside the cell like it was labeled in that animation. The role of oxygen, again, is the final electron acceptor. If you don't have the oxygen to accept that electron, you have to go to an anaerobic metabolism as opposed to aerobic, air being oxygen. It doesn't work very well, and it certainly doesn't work very long. I mean, if you could find a way to have this go on, to have another electron acceptor in there besides oxygen, then you wouldn't have to breathe. At this point, obviously, we're not there. ATP balance sheet, I'm going to let you go through that in your book. Here it says theoretical ATP yields 36 to 38. If you look at the actual number, there is some ATP that's used to drive the proton pumps to provide other energy needs between the Krebs and the electron transport system, so it's really 30 to 32. And that's detailed in your book also. Okay, this is out of the book, so this is the 32 to 34. Okay, metabolisms of energy and lipids and proteins. They use the same pathways used for metabolism of pyruvate. Like I said, pyruvate. Anytime everything goes back to pyruvate, pyruvate is broken down in the mitochondria. Your acetyl-CoA goes through the Krebs cycle, and bada bang, you've got energy. The only thing that's a little bit different is your fat metabolism, where it comes right in as acetyl-coenzyme A. It goes right into that. It doesn't go through pyruvate. Okay, too much energy. When you take more food in, you only store so much ATP, and it's only a few grams. It's not that much. So your body, you store 80 to 90 grams of glycogen. The rest of it is stored in fat. So when your adipose cells, your fat cells say, hey, I got all this extra glucose, I've got all the ATP that I need, I'm going to make fat. And it stores it in fat. Okay, too much energy. You have glycogen, glucose 1-phosphate. If you go down, you're breaking it down. If you go up, you're making glycogen. Glycogen goes in the glucose 1-phosphate. Now in the muscle stores glycogen. The muscle stores glucose 1-phosphate. The muscle can break that down into energy. What the muscle does not have is a glucose phosphatase. The muscle does not have the enzyme to break the phosphate off that glucose. With that phosphate attached to the glucose, it can't get out of the muscle. So it can stay in there and get used aerobically or anaerobically and end up getting turned into lactic acid. But it can't get out into the system. The only organ that has the enzyme to break that phosphate off is the liver. And the liver can actually just shoot glucose out into the bloodstream. To the brain, to the heart, especially to the brain, because that is really the primary energy source for the brain. But again, you go down, glucose phosphate, glucose can go back and forth there. It goes to fructose. It goes to phosphoglyceraldehyde. Then if you have enough, you don't need it in the Krebs cycle, it goes to phosphoglyceraldehyde, then it's changed into glycerol. The glycerol is changed into fats. You get 3 glycerols put together to form a triglyceride, or else they go into a long fatty acid chain, and you have fatty acid. Okay, lipid metabolism. ATP rises after an energy-rich meal. The production of ATP is inhibited. Glucose doesn't go through glycolysis. It's formed together. Pyruvate and acetyl-CoA are already formed or joined together to produce a variety of lipids. Remember, your pyruvate is a 3-carbon. Your acetyl-CoA is 2-carbon. So now you've got your 5-carbon ring. Remember, your lipids, your steroids, your cholesterol all have that 5-carbon ring on them. Fatty acids combine with glycerol to form triglycerides. In the fat tissue, adipose tissue, and liver, it's called lipogenesis, generating lipids. Again, a very efficient way to store energy. Not that I like it very much, but an efficient way to store energy. Acetyl-CoA to lipids. This just gives you, you've got the acetyl-CoA, it can form triglycerides, it can form phospholipids, it can go into the Krebs cycle, it can go into cholesterol, bile, steroids. It's a building block for a lot of substances. White fat. That's your baking fat. A great way to store energy. 1 gram of fat is 9 kilograms. Excuse me. 1 gram of fat is 9 kilocalories of energy. Lipolysis is the breaking down of triglycerides using the enzyme lipase. Fatty acids can enter the blood and be used for energy elsewhere. I mean, you get somebody after a bunch of ice cream, depending on how their system is, that serum, that blood just, I mean, it kind of looks like ice cream. It's that white and fat, all those fat molecules, they're called chylomicrons. Fatty acids as an energy source, beta oxidation. Enzymes remove the acetic acid molecules from the fatty acid to form coacetylate. Acetyl-CoA. So it's chopped off in 2-carbon chains. And then the beta oxidation turns it into acetyl-CoA. Acetyl-CoA goes right into the Krebs cycle, like we showed you. Attaches to the 4-carbon carrier and right around the Krebs cycle. 3 NADH, 1 FADH, and 1 ATP. But for a 16-carbon fatty acid chain, that happens 8 times. In the end, that's 108 ATP that are formed. So what I'm trying to get across is think back to the Krebs cycle. The glycogenesis gives you pyruvate. The glycogenolysis, the protein breakdown in that, gives you pyruvate. The fatty acid breakdown gives you acetyl-CoA. So the pyruvate from the glycogenesis, the pyruvate from the protein breakdown goes to acetyl-CoA. From the fat, it goes directly to acetyl-CoA, which is right at the start of your Krebs cycle. Okay? So try to simplify it. You know that acetyl-CoA is going to get to the Krebs cycle, go through the Krebs cycle. After the Krebs cycle, it goes through the electron transport chain. Everything else, you break down a protein, you break down a fat, you break down a sugar, in some form or another, is going to give you acetyl-CoA. And then it goes through. Think about it. Okay? Take a deep breath. Think about it. Pyruvate goes to acetyl-CoA. Fats go to acetyl-CoA. Everything goes to acetyl-CoA. When it gets to acetyl-CoA, if you need energy, it goes through the Krebs cycle. If you have too much energy, it's formed back into fat. And your fat's turned over anyway. Your proteins are turned over. So this is where I'd like to say, don't make it harder than it is. But
What do you mean your fat's turned over and your protein's turned over?
your body's always turning it over. It's breaking it down and rebuilding it all the time.
Puts new fat in place of old fat,
Yeah.
or just keep the same fat?
No, new instead of old. Yeah. Your amino acids, we'll get into a little, well, we might not. Beta oxidation. So this is just the breakdown of fat into acetyl-CoA that goes into the Krebs cycle. And there's your kind of an accounting. This is in your book. Brown fat, it's involved, it's rich. They didn't think they had it in adults for a long time, but we still have some. Usually found in newborns. It causes thermogenesis, heat production, because you don't have the muscle mass to keep you warm. Ketone bodies, as you break down fats. As you break down fats, the body produces ketones in the liver. And if you are breaking down, and then from ketones into acetyl-CoA, but if you're in starvation and if you're in a diet, you're going to break down more fats, then the liver is going to convert into acetyl-CoA, and those come out in the body. Those come out in the urine. If they build up, they circulate in the blood. If they build up too much, as in diabetes, where you are breaking down all the fat because you can't get any sugar into the cells, any glucose into the cells, you get ketosis or ketoacidosis, and at some point, if they build up too much, they can kill you. Like I said, I see patients in the ICU with diabetic ketoacidosis all the time. They're off their insulin, they come in with blood sugars of 900 to 1,000. Their ketone bodies are building up because their body hasn't got any insulin to get glucose into the cells. It's turned to a fat metabolism to get energy. The byproduct of that is ketone bodies.
Would the same thing happen if you're not diabetic, but you just are starving?
Yes, exactly. But usually, if you're starving, you pretty much used up a lot of it. I don't see it that high in somebody. I don't see a DKA or a ketoacidosis, not to that extent in a starving person versus a diabetic person who, when they have glucose, when they have insulin, is putting all this fat on. It's very easy for them to put fat on.
Because they're intaking calories.
Yeah. I mean, the fact that the insulin is pushing the glucose into the cells so fast, their blood sugar, you always feel hungry. And they tend to gain a lot more weight, which is just compounds the problem of being diabetic. But they've got a lot more fat generally than a starving person to turn into ketone. I don't know that I've ever seen anybody in the hospital with ketoacidosis that wasn't diabetes. Amino acid metabolism. We can break down amino acids from the dietary proteins. Again, these amino acids are they have a nitrogen. They can be broken down, they end up in carbohydrates, which ends up in pyruvate, which ends up in acetyl-CoA, which goes through the Krebs cycle. The nitrogenous bases are recycled. This goes on all the time. If more amino acids are consumed than needed, the excess are used for energy or converted into fat. But you do need them to replace proteins. If you look in the book, you have what you call essential amino acids. There's 7 or 8 of them. The body cannot produce them. I'm not going to make you learn the list of essential amino acids, but it's probably going to be a good test question as to know what we mean by essential amino acids. Essential amino acids. Bodies can make 12 of the 20. 9 in children. I'm not sure what the 9th is. I'll find out. But you've got to have those in the diet. You've got to take them in. Histidine. in children. This is a table out of your book. Transamination. The body can take pyruvate and Krebs cycle intermediates, and they can convert them into amino acids. These are the ones that the body can make by adding the amine group or the nitrogen group. A lot of the times they'll obtain them from other amino acids that are being broken down, and it's called transamination. It takes place in the liver. It requires vitamin B6. When we look for hepatitis, when we look for liver problems, we use two enzymes that we look for increasing is ALT, alanine transaminase, and AST, aspartamine transaminase. If you have liver problems, these build up, they get into the blood. Here's AST. It's taking glutamic acid and oxoacetic acid and turning it into aspartic acid. That's it, aspartic transaminase. Alanine transaminase, again, takes glutamic acid and pyruvate and turns it into alanine, into your amino acids. Oxidative deamination, if they're more than you need, the amine groups are stripped, they're excreted as urea. If they're not excreted and the liver isn't working right, they form ammonia. Ammonia is extremely toxic. And if your liver isn't working right, you get ammonia, and that's not good. That's generally very much end-stage liver disease. Oxidative deamination. You look at the alanine. It's oxoacetic acid plus pyruvate. Well, if we break down the amino acids, we're back to getting pyruvate. Pyruvate goes to acetyl-CoA. Acetyl-CoA goes into the glucose, into the Krebs cycle. If you have a lot of extra, they make convert it to glucose because you get to the acetyl-CoA. If you've got too much energy, if it doesn't go through the Krebs cycle, if you look at that chart, it goes back and it's converted into fat again. And glucose neogenesis, it looks like it can go through the Cori cycle. I wasn't aware of that. If we need to, we'll come back. That should just about do it. Amino acids as energy, I think we've gone through that. It's citric acid, acetyl-CoA, going back into the Krebs cycle again. Different energy sources. Glycogen, glucose, this again is in your book, we've talked about this. Protein, fats, glycogen, glucose. This is just what organs use them. Brain uses almost entirely glucose, a little bit of ketone. Skeletal muscles, fatty acids. Like I had said, if you build up, as you work out, you build up the ability of the skeletal muscles to use fatty acids. Livers, fatty acids, hearts, glucose, and fatty acids. Have a nice evening. We got through chapter 5. Chapter 6 is actually relatively short and relatively simple. We'll probably try to tackle 7 and then part of 6 and 7 on Tuesday and finish off 6 on Thursday because it very much coincides with what we're doing in the lab. Have a good weekend. Try to stay warm. It's going to be extremely cold this weekend. They say it's supposed to snow in San Francisco. Down to sea level. Well, extremely cold for California. I'm from Minnesota. So, if you can't tell from the accent, I don't have it.
Oh, no, that's fine. Oh, I have a CD that I just go through.
You do? Okay, Brian.